So thank you, Luis, for the introduction. It's a pleasure to be here. I know a lot of faces. I know some names. I know some very famous Twitter accounts that work here at Michigan. But I hope I get to meet more of you. So as Luis said, I was trained in physics, but we're doing things that I think some people might call ecology, some people might call evolution. So I'm going to tell you a little bit about our work on the antibiotic response in Enterococcus faecalis. So this is a gram-positive bacteria. It's a big cause of nosocomial and hospital-acquired infections. So the main question we want to ask is, how do population dynamics, and I mean populations of bacteria, not populations of people, how do populations of bacteria contribute to antibiotic resistance? What are those population dynamics? And so there's some essential information that we have to know, and this is one of, I'd say, sort of the classical ways to address this problem, is you zoom in on a cell, right, and you ask, what's inside of that cell? What's going on to make this cell resistant to a drug? So one possibility is that you make an enzyme. This particular enzyme is called beta-lactamase. That enzyme renders the drug ineffective. Okay, so under this situation, the main goal is to understand the molecular mechanisms giving rise to resistance. So of course this is essential, right? But it's entirely incomplete, I would argue. And I think I'm probably preaching to the choir here a little bit, right? Because bacteria don't live as isolated cells. So maybe we need to step back and try to observe the community behavior. And so just one example that you can come up with is imagine just two of these guys make an enzyme. Okay, and this enzyme that's made by these two guys degrades the drug, and it sort of cleans up the local environment so that other guys who don't make this enzyme can survive. This is a classic example of cheating, and a lot of game theory applications you can imagine could come from something like this. There's a beautiful paper, Molecular Systems Biology, from Jeff Gore's group at MIT on this particular topic. My point is that the population behavior can be really, really rich, and our goal is to try to understand this population level behavior. And so a lot of the tools to understand this population behavior, in my terminology, I call statistical physics. Now, of course, I think there are many of the same tools in ecology and evolution. So what matters in statistical physics? Well, it's the interactions between particles that typically matter, or between units, between constituent parts of a system. This is just a movie of, you've seen these beautiful movies of birds flocking. Right? And so if you want to understand something like this, of course you have to know how birds fly, but what you really need to know is how one bird changes his motion relative to the other bird, right? So it sort of depends on the length scale you want to try to describe. There's this video that's maybe the most interesting part of my talk today. If you look up this video, when this phone rings, the ants circle it, and when it stops ringing, they stop circling it. So I'm just going to leave that because I don't understand that one. But I think that's one of the most interesting things I've seen. There was just something that went around Twitter, I think yesterday, that I meant to grab, of sheep viewed from above and how it looks basically like a complex fluid and does all these sheep do all these crazy things. You can even apply something like this to the Supreme Court. So there's a paper called The Statistical Mechanics of the U.S. Supreme Court. And basically what they're trying to do is predict how the Supreme Court will behave, and they took a lot of historical data. And what they found is that if you know sort of the individual leanings of each justice, you know if they lean left or right, you can't do a very good job of predicting the outcome that the Supreme Court comes to. But if you account for correlations between their decisions, you can describe it really, really well. Okay, so they actually developed what's called a maximum entropy model that many of you probably know about. It takes into account pairwise information, but not higher order correlations. We developed something when I was a postdoc very similar to describe the way antibiotics affect populations of bacteria. Okay, this is just in a lab setting. But if you apply one antibiotic, it kills the bacteria. You apply a second one, it also kills the bacteria. Sometimes when you put them together, they don't work anymore, right? The population survives. And it turns out that it's very hard going from single drugs to two drugs to predict what happens. But if you know the pairwise interactions between drugs, you can predict higher order combinations very well. So it's the same type of thing. Same laws are governing the Supreme Court and antibiotics, maybe. So, of course, there's a lot of powerful mathematical tools. These are the books, you know, that someone in my group or with my background is very familiar with. You know, books in statistical physics, stochastic processes, bifurcation theory, dynamical systems. Many of you probably know these books. Others may know many of the same techniques that were applied sometimes earlier, sometimes later, sometimes with different goals in ecology and evolutionary biology, right? So hopefully there'll be some overlap here, at least in interest. I may call it statistical physics when it's really ecology, but that's sort of my background. So one of the goals, you know, we wanted to do when we started setting up the labs, we wanted to be able to measure quantitatively growth of bacterial populations. And so we built something that was discovered, you know, or first built 50, 60, 70 years ago, something called a chemostat, right? But we tried to make it a little fancier, and all of you, or many of you know what a chemostat is. It tries to keep the environment constant. And we had multiple papers that were inspiration for this. We borrowed some of their techniques if they worked and adapted it as we needed. But this is an example, right? So it looks really messy, so that's good, right? It's a good solid experimental system. Each of these is a vial. So there's me holding the vial. You can't see my fingernails a little dirty there. I've been self-conscious about that, but there's not a good enough resolution here. So this is just a vial. This is where the bacteria grow. At the top of the vial are silicone tubes that come out. You connect these silicone tubes to peristaltic pumps. Each of these pumps, there's up to 6 of them per vial right now. Each of these pumps you can control with a computer, just a voltage output from a simple data acquisition card. And then on the side of these, we drilled holes on the left and the right side at some angle, and you put little LEDs, right? So there's an infrared LED and a detector. And the LED flashes, and the detector measures how much light reaches the detector. And it turns out if you build a little circuit and you measure the voltage from this output, that this voltage corresponds very well to optical density that you can read in a big, fancier spectrometer, right? It's just essentially light scattering. So this gives us a way to at least in many situations, measure population dynamics, measure the density of cells. Now, in principle, we can do a lot of other things with these, and we're just really starting to play around with this. For example, we can build habitats that are spatially heterogeneous. So maybe we have one vial with a small amount of drug, one vial with a second drug, and a third vial that's got a larger amount of drug, right? We can create spatial heterogeneities over space, and then we can create flows or patterns of migration between these that are all computer controlled in whatever arbitrary way we want, okay? So these are really, really simple instruments. If any of you guys are interested, I can show you. I have a graduate student, Jason, who put a lot of this together. He can build anything, so you should talk to him if you're interested in this. But before we get into anything complicated, we just wanted to start simple. So this is the first thing we did with this, is we said, okay, we want to claim that the response to drugs is some type of a community phenomenon. So the first thing we have to say is, wouldn't that mean then that the behavior should depend on the population density, right, which could scale sort of the length scale of the different interactions? And so the answer to this, when you look in the literature that many of you may know, is yes, there's something called the inoculum effect. And the inoculum effect says that if I start with a larger inoculum of cells, a larger starting fraction, a starting population, called a bigger inoculum, then the inhibitory concentration of drug required to kill that population is larger. Okay, so there's this relationship between your starting population density and how much drug it takes to kill the cell. And so it's a little difficult to interpret this, though, and one of the reasons is that these measurements are performed on growing populations, and the populations can sometimes change density by orders of magnitude on the time scale of one of these experiments. And so it's difficult to say, what is the relationship exactly between population growth in the presence of drug and density, right? The other thing is it's really just a binary output, the cells survive or not. We'd like to get somewhat of more quantitative output that we could use maybe to build mathematical models, okay? So it turns out that's really easy to measure in one of these instruments. Our model system is Enterococcus faecalis. It's a source of hospital-acquired infection. What we do is we use feedback to control the cell density. So this red is just a growth curve. It's a typical growth curve. The blue one is a growth curve where we said, okay, hold it at a particular density here, right? And it's very simple. All we do to hold it there is we pump in fresh media and we pump out some of the cells. And we can keep track of how often that pump turns on and off, or the flow rate of that pump. From that, we can estimate the per capita growth rate of the population. Okay, so we have a real-time measure of growth rate as a function of cell density and anything else that you put in the environment. So the experiment's really simple. We just measure growth at a constant drug concentration, and then we do this as a function of different densities. We do this on short length scales, several hours is this experiment. Okay? So this is what an experiment might look like. This is the per capita growth on the y-axis. This is the time on the x-axis. We add drug at time 0. There's this sort of gray region where there's transient effects. We don't know what's going on here, although we did see there was recently a paper that built a mathematical model for this particular class of drug and predicted this exact dynamics. Kind of interested in looking into that more. What we care about here, though, is the steady state dynamics, which is in the white region. And each of these curves has grown at a different optical density, right? So a different population density. But they all have the exact same concentration of drug. And so what you can see very clearly is that the drug efficacy strongly depends on the density. And so you can plot something like this. You can measure cell density. This is the growth relative to untreated cells, and you get a curve like this. And this part here really measures how big this density effect is. Okay, now one thing I want to point out here is, One thing I want to point out here is that this is just a growth curve. We're in the exponential part of growth, right? So if we take away the drug, these cells are growing the same. And this is the exact experiment with no drug. Like, there's not much change in the growth rate in the absence of drug. Okay, so this is all happening in the part of the bacterial growth phase where you would say everything is sort of exponential, everything's sort of growing nicely, right? But we still see these really strong density effects. And so we did this for a lot of common antibiotics. So this is a plot with 9 different ones. The main point I want you to sort of take home is that these effects are not very rare, and that there are sort of multiple types of these effects. Most of them show an increase in growth as a function of cell density, but occasionally they'll show a decrease in growth. This is for ampicillin. Okay? So this was sort of mysterious to us. And so, of course, one of the most interesting questions is why, right? But there's a lot of different drugs here. There could be many different mechanisms. And if you look in the literature, there are certainly mechanisms out there that could describe something, or that could underlie something like this. One would be enzyme degradation of the drug. As far as we know, they are not making an enzyme that degrades the drug in these cases. There's a paper on heat shock and growth by stability. It's a more complicated mechanism. We know at least one of ours corresponds to that. We did some tests to show that. You could also imagine the drug is just being sequestered inside of the cell, and so it's effectively lowering the concentration of drug everywhere else. We did find one additional mechanism that described 2 of the cases. This mechanism is also very, very simple. As the community grows, the cells divide, and what happens? Cells metabolize environmental sugars, and the environmental pH drops. When the pH drops, there's a lot of literature that points to the chemistry of the drug essentially changing and acting very differently. So there are at least 2 drugs where we can point to the change in pH leading to the change in the effective density dependence. But we did this for a lot of drugs, and for many of the drugs, we still don't know the mechanism. Okay, but what's kind of nice about this is even without the mechanism, you can still make some predictions. So I'm going to walk you through how you can make some predictions. So we built a really simple mathematical model of our turbidostat, so that's a constant density chemostat. So we wanted to write a simple equation for the drug concentration in the vial. So there's drug in some reservoir. That drug's being pumped into the vial at some flow rate F. The volume of the vial is V. And then there's flow out that contains both drug and cells, right? So we can write a simple continuity equation for the drug concentration in the vial. Depends on the flow rate F, the volume, the concentration over here in the input reservoir, and the drug in the reservoir itself. Okay? Now, we try to account for density dependence effectively by adding a term that is just a per capita rate of drug decay. Okay? So this is a parameter, we're calling it epsilon here. Okay? It just gives you something, it gives you some information about the time scale and the magnitude of this density effect. And we can simplify this equation. First of all, we'll look in the steady state, so we'll set this equal to 0. We also know a little more because F over V in a turbidostat has to be G, the growth rate. So we can plug in G there. So at the steady state, we get a little bit simpler equation. We're not completely finished, though, because the drug concentration also feeds back on the growth rate. And that's something we can measure independently at very low densities. And this is just a growth response curve measured at low densities. And so we can estimate what the shape of this function looks like. Right? It typically looks like some type of sigmoidal binding function. We can invert that function, and now we get a nonlinear equation, so we can't solve it analytically. But the equation relates inputs, which are drug in the feed chamber, and cell density, to the per capita growth rate that we measure. And the only free parameter is this epsilon, which we can then estimate from our experiments. Okay? And so epsilon turns out to be really powerful because it gives you information about sort of the relationship between the magnitude of the effect and how fast the effect happens. So now you can start putting this into mathematical models and say things like, so does this really matter for a growing population? If the effect's really small, but it happens really quickly, maybe it matters, right? Or if the effect is really, really large, but happens on a very slow time scale, it could matter. In other regimes, it might not matter, right? But we can test this. So the first test we did was really simple. We just said, imagine we have a logistically growing population. We have growth of that population that's a function of the drug. Again, that's something we can independently measure. You have a carrying capacity, which we can independently measure. And then all we do is we add to this model the density dependence that we measured. Okay, so we allow the drug to decay according to this density dependence. And when you add this in here, you can say, okay, let's start with some concentration of drug and ask how long it takes the population to reach its threshold size, maybe to get to stationary phase or something like that. Okay, we picked something close to stationary phase. And so here's an example. So here's an example of the line, which is just a simple theoretical prediction. This is the concentration of drug, and this is the relative time it takes to go to threshold. Right? So qualitatively, there's nothing surprising here. As you use more drug, it takes longer to get to stationary phase. And then we did the experiments, and it's nice, the theory predicts it pretty well, so that's fine. One thing that's really nice about this, though, is we did this experiment with the drug that is, whose density dependence relies on pH. Remember I told you there's this one mechanism of pH. What that means is we have a dial to turn the density dependence off. If we use really strong buffer, for example, we can turn the density dependence off. And so we can do an experiment now that say, well, how much of this is due to the density dependence itself? So it's very easy in a model. In a model, we can calculate, if this density dependence goes to 0, what we expect, and then the dots are the experiments themselves. And so the density dependence can be fairly big, right? A factor of 2 or so. And this is in, growing populations in a lab setting, right? So they're growing pretty quickly. So the density dependence are fairly large. And then we said, well, what about if we take a clinical model? We take some type of pharmacokinetic, pharmacodynamic model where the drug is being, dosed periodically every day or every 8 hours or something like this. And we ask now, in this case, the drug's decaying because of some type of metabolism in the body, not due to the density dependence. But we add to this model the density dependence that we measure with this parameter epsilon. And then we ask, what's the long-time behavior? Can you clear the infection? This is a really simple model. You can, we essentially average over fast time scale dynamics, and you can derive an analytical phase diagram that looks like this. So these red curves are stable fixed points. The dashed curves are unstable fixed points. The y-axis is the population density. So this is essentially the starting population density of the model. The x-axis is the initial antibiotic concentration. This blue region here, or the region above the dashed line, is the region where the infection is cleared. This region down here is where the infection is not clear. The shading just represents numerical results. So what you see here is if you start at very low drug concentrations, the infection's always cleared, right? Not surprising. And if you can use a whole lot of drug, the infection's never cleared at low drug, right? At very high drug, the infection is always cleared. But there's this interesting region in the middle where the result is bistable. So what that essentially means is if your population density is above some critical threshold, the treatment won't work, the population will survive. If it's below that threshold, the population will decay. And you can put in numbers here based on the epsilons we measure, and it looks like over some typical density ranges for infections, you can see that treatments at something like 100 times the MIC could fail. Now, it's important to note that this would be failure without genetic resistance, right? This is a phenotypic resistance. If we start at this high density and we see that they grow really well on the drug, then we lower the density down, the effect goes away. Right? This is a phenotypic change. So you can read more about this if you want to. I think that's all I'm going to say about this particular thing. There's a paper in PLOS Computational Biology on it. Now, if you start looking at this on longer time scales, you can keep these experiments going, and then what you start to see is genetic resistance pop up, right? And this is what it would look like in one of these instruments. This is just your growth rate relative to untreated cells. It's essentially related to the pump rates needed to keep the population constant. And this is in some constant concentration of drug. I think this is ciprofloxacin. It's not particularly important. The amount of drug keeps the growth at about 20% of its typical growth rate. And then somewhere around 10 or 12 hours, you see that growth rate spike up. And you can pull this out and see that it's, in fact, a resistant mutant. Okay, so over longer time scales, you could watch resistant mutants pop up in this way. And you can also mimic more complex spatial meta-populations that are connected in various ways. So you can imagine doing something where the drug concentration is say, homogeneous over space, or has a peak in the middle, or maybe has a valley in the middle. And you can see how that affects the length of time it takes for resistance to take over this population. And this is actually a pretty general problem, and a student of mine, Max from physics, is working on this problem from a theoretical perspective using some tools from statistical physics. And the question is just how does spatial heterogeneity and selection pressure impact resistance to evolution? So this is a very widely studied problem. Max has a nice simple model that we can make semi-analytical conclusions with. But of course, selection pressure is not just, it's not just density or drug concentration. It could also be things like temperature or pH or some type of heterogeneity in the host or immune response, or you can imagine multiple other things. I'll tell you only that in theory, the answer is that heterogeneity can speed up evolution or it can slow it down, and it depends on the details, right? So that's something that's coming soon. Hopefully, I'm not going to talk much more about it now, but I wanted to at least mention it. Hopefully, some experiments too. Any questions? Okay, so some of you might say, well, that's fine, but bacteria don't really live in these well-stirred environments, right? This is a pretty artificial environment. It's nice, it gives us quantitative results. Maybe it's not really how things happen in nature. So what happens at this really high density limit where biofilms form? And so many of you guys know about biofilms. These are these sort of densely packed surface-associated communities. Some things I want you to know about biofilms is that they can be extremely heterogeneous. They can be genetically heterogeneous. They can be phenotypically heterogeneous, even in a genetic population. And there's lots of sort of complex behavior that can come from this. Another thing that can happen is that these guys are often more resistant to drug. And at least one explanation for that, although it's not the only one and it's not always true, is that the biofilms don't always, the drugs don't penetrate the biofilms well often. So mostly you think of a biofilm as something that's even harder to kill, and that's a problem because these are things that grow on heart catheters or medical instruments, right? So these are even more difficult to kill. And the particular species we look at grows biofilms by doing something kind of funny, and that is that it requires there to be extracellular DNA around. And so it's believed right now that this extracellular DNA plays a role in cell adhesion and structure of the biofilm, right? And it's something that's been found in other species too, particularly gram-positive species. And the main source of this eDNA is thought to be either autolysis, the cell sort of exploding, or autolysis from either altruistic suicide, the cell's exploding for the good of the community, or fratricide. Right? And so the question we wanted to ask, given this kind of unique situation, is, well, there's something at least to us seemed kind of obvious, is, well, what happens if we add an antibiotic to this, right, which could potentially affect the rate of cell lysis? And so this is work that a graduate student, Win Yu from physics and Kelsey from biophysics, worked on over the last, I don't know, six months to a year. So we did another very simple assay. We measure biofilm formation using a crystal violet assay. This is a common assay for measuring biofilms. Basically, you grow them in plates. We use 96-well plates. You let them grow for 24 hours, and you stain them with a crystal violet, just a type of cell stain. You then rinse them and solubilize it, and it's kind of this tedious process, but once you finally do it, you can essentially quantify the size of the biofilm using a plate reader. Okay, so this is a very standard biofilm assay. So what we did is we did this assay, but we added ampicillin, a cell wall synthesis inhibitor, during the formation. And we saw something really interesting. What we saw is that as a function of ampicillin at very low concentrations, the amount of biofilm actually increases. So low amounts of ampicillin are actually increasing the amount of biofilm. So this is counterintuitive at first. Antibiotics are supposed to get rid of the biofilm. In fact, biofilms are even harder to kill than planktonic populations. Here it looks like, at least at these very low doses, we're seeing an increase in the biofilm. So we said, well, that's fine. We did it in two different types of media, BHI and TSB, but this is only one strain. So we tried another strain. Is this specific to that strain? No, we see it there too. It's a little harder to see. My colors are bad, but we see it in other strains too. Okay, so we said, well, that's fine. What about other antibiotics? This is only one drug, right? So this is a cell wall synthesis inhibitor. We looked at other classes of drugs. When we looked at other inhibitors of cell wall synthesis, we saw something similar, an increase in biofilm at these low concentrations. And then what we found was really, really interesting is when we looked at other types of drugs, protein synthesis inhibitors, those are in red, DNA synthesis inhibitors, that's the middle column, RNA synthesis or folic acid synthesis inhibitors. In all of those cases, we didn't see evidence of this increased biofilm formation. So we said, okay, that's interesting, right? We see some type of order, which is always nice when you're doing an experiment, right? And so we had a hypothesis. We said, maybe what's happening is these cell wall drugs, which are known to increase lysis even at low concentrations, maybe what they're doing is they're causing an increase in lysis and in turn an increase in this extracellular DNA, and that's leading to, in some way, a more stable biofilm formation process. So first we had to ask, was lysis occurring, right? We know if you look in liquid cultures, this is a dose response curve, so this is the population density relative to untreated cells as a function of drug, and we're up here in this regime, right? So what that means is on liquid cultures, we're at a drug concentration that really doesn't do much, right? It doesn't affect viability much at all. And so the first question was, do we even see lysis at these concentrations? And that's something we can measure. So we measure this again with a standard assay. It essentially measures ATP in the surrounding media as a function of drug. And we saw that, yes, there is this dramatic increase in lysis. Now, this is about where the peak is, so the increase is a little less than twofold in that region. Okay? But there is lysis occurring. And then you can also measure the eDNA, right? You can kind of scrape the biofilms extract DNA, and then you can run it out on the gel. This time you extract DNA from once you've removed the cells themselves. This is just a picture of the gel, but this is the important thing in the middle is just a quantitative measurement. And what we see is as a function of ampicillin, we also increase the amount of extracellular DNA. Right? We also checked RNA, that increases, but at higher concentrations, it looks like. Okay? So the eDNA also increases. So this is at least consistent with our hypothesis. And then we said, well, maybe we're just somehow increasing the non-living cell mass in the biofilm. Maybe it's not actually living cells. So we used confocal microscopy with a live dead cell stain, and we measured biofilms in 3 dimensions at different concentrations of ampicillin. It's a little hard to tell on this figure. We were convinced from looking by eye, but of course we did this more quantitatively. So you can see maybe an increase in the density here as you go left to right, and then it's going to decrease again here. So we did this for many biofilms grown on different days, but the same conditions, many replicates, many slices. We averaged over something like 10 to the 5th cells total, and we got a plot that looks like this. So the number of living cells in the biofilm is in fact increasing as a function of ampicillin, and then starts to go down at 0.2, so that's consistent with the bulk results. The number of dead cells is monotonically increasing in this range. It's not super surprising. But it's much, much smaller than the total number of cells. So then we said, okay, well, what about if we induce lysis chemically? Right? This is an antibiotic, but antibiotics do all kinds of things, right? Can be very complex cellular responses. What if we use something simple, some type of detergent or something, and we try to induce lysis that way? And so we did that. So we added just a chemical inducer of cell lysis, and we saw something very similar. The amount of biofilm formation goes up at low concentrations of this lysis inducer. This inset is just, we can actually measure the lysis. We see that it actually does go up. And the size of the effect is about 20%, which was about what we saw with the antibiotics. Okay, so again, this is consistent with our hypothesis. And so we sort of tried to put this together into a conceptual model. The idea is that you have a big biofilm here. You have lysis that's releasing the extracellular DNA. When the extracellular DNA is released, it in some way facilitates attachment or stability of that attachment once it occurs. And this attachment process occurs with some rate C, and it depends on the number of planktonic cells in the solution, right? You have to have living cells around in order for them to attach. And then there's also a term that basically just says there's a decay of this extracellular material over time. Once a cell dies, it only sticks around for a certain amount of time. And what's important about this is you have 2 main ingredients of this model that work against each other. Lysis is we expect to increase with drug, right? But the number of planktonic cells we expect to decrease with drug. And so what we think is that there's some trade-off going on between these 2 effects. And it turns out that we already have the functional forms of these 2 effects, right? We know how planktonic cells change as a function of drug, and we know how lysis changes as a function of drug. Okay? So we can put this into a very simple mathematical model where you have logistic growth, you have some cell death term. D is now our living or our dead cell mass, so it's like eDNA if you want. We have some detachment term, and then what's key is you have this attachment term, and the attachment term again depends on the lysed cell material. Right? So this couples the lysing of the cells to the increase in the mass of the biofilm. And you can analytically drive different things. It turns out some of our measurements show that some of these parameters are very small, so you can use simple type of perturbation theory to estimate things like the peak height, how big this height should be. You can also drive things like the location of the peak, right? And they all turn out to be dependent strongly on this parameter we call a coupling parameter. I'm not going to stress too much about what that means because I think there's an easier way to think about it. You can also derive a phase diagram from this model. This phase diagram includes some region where there's enhanced biofilm formation and a region where there's decreased biofilm formation. The 2 axes are the amount of lysis on the horizontal axis and the amount of planktonic cells. Each of these axes depend on the amount of antibiotic. So as you start with 0 antibiotic, you are somewhere in this phase space, and it turns out you have to fall on this line. Right? So when you start with 0 drug, you live somewhere on this line, and what determines whether or not antibiotic increases or decreases, according to this model, is the path you take as you add drug. Right? These are, again, 2 functions that both depend on drug, so this is a parametric curve through this space. And you can imagine 2 different cases. Curve A would give you a peak something like this, so this is like what we saw for ampicillin. So what it's saying is that the lysis is in some sense, the lysis is the x-axis, is in some sense happening faster than the planktonic cells are decreasing. Right? So the amount of lysis is increasing faster than the planktonic cells are decreasing as you add drug. When I say faster, I mean as a function of drug, right? Not time. However, you can imagine a different path through this space that looks more like B, and in this case, the lysis is in some sense slower than the planktonic cell death. Okay? So it's a pretty conceptually simple model that shows how, as you add antibiotic, this interplay between cell lysis, which increases with drug, and planktonic cells, which decreases with drug, gives rise to increased biofilm formation. And then, you can do a lot of analysis of the model, but one of the most important things with a model, of course, is to try to test it. And so we said, okay, well, what happens if we add DNase to degrade the extracellular DNA during the formation process? Well, that means this parameter would be increased, right, the decay of the extracellular material. We can easily plug that into a model and look qualitatively for what we expect, and we expect the curve to decrease in size, and we expect a slight shift in the peak of the concentration. So we did this experiment both with microscopy and in bulk. We saw roughly qualitatively similar agreement. You could also ask, well, what happens if we add a second antibiotic, but one that doesn't induce cell lysis, right? So in this case, you might expect in our model for the number of planktonic cells to just drop by a constant, right? But no increase in the lysis. You can put that into the model, and you see something that looks very similar. Okay, and we did that experiment too. Each of these bottom curves are with a second non-lysing antibiotic added. We see something, again, that's qualitatively similar to what you predict. We can also ask, what would happen if you add a chemical that inhibits cell lysis? And luckily, there were some of these out here in the literature that have been shown with gram-positive bacteria to decrease cell lysis. First, we measured to see that it actually does decrease cell lysis, and it does. Then our model would predict if we decrease cell lysis, it means the lysis function is basically decreasing by a constant. And now you get something kind of surprising that happens. When the lysis decreases by a constant, the relative peak of the biofilm formation actually goes up, and it shifts to higher drug concentrations. Okay, so we can do this experiment, and we see at least a very clear increase in the peak. Right? You can argue whether or not that's actually increasing. It depends on how big the effect is. Right? So this is cool. We were happy about this. We have a model. We have sort of a conceptual framework. We've tried to sort of perturb the model a little bit, and it seems to hold up. Okay? So if you want to read more about this story, there's a paper that just came out. I would tell you that this does not include, this is not a preprint, but right after it's accepted, a proof, basically, version. So if you want to read sort of the details, the SIs on the bioarchive. I don't know if you guys like this, but I recommend it. So I've still got, this goes till around 5? Okay, cool. So you might also ask then, from there, what happens if I use much higher drug concentrations, right? This is low enough drug concentrations that these sensitive cells, not only surviving, they're in some sense thriving. Okay? Well, if I use a lot more antibiotics, first of all, if I only have sensitive cells, nothing interesting happens. It kills them. Right? We did that experiment. It's not exciting. What might be exciting, though, is to consider a case where there is genetic resistance in the population. So let's say we have a mixture of genetically sensitive and resistant cells, so we can engineer various types of resistance. We want to say, okay, I have some small number of resistant cells in red, and as this biofilm forms, how does this spread in the population? And not just how does it spread, but what happens to the spatial positioning of the cells as this happens? So, just for details, our resistant cells constitutively express a beta-lactamase, so an enzyme that breaks down the drug. They hold on to this enzyme. But you can imagine still there could be some cooperation happening here. Right? Okay, so what do we see? Well, we did confocal microscopy. We developed some simple imaging algorithms that allow us to find all the cells and their locations and then do statistics on sort of where these things are. And there's only really one thing I'm going to show you from this, but I think it's pretty cool. So if you pick a sensitive cell, so I pick one of these blue cells. This is basically the analysis we did. You pick one of these blue cells, and you say, within some neighborhood of this blue cell, what fraction of my neighbors are resistant? So I'm zooming in on the sensitive. I'm saying, is your neighborhood resistant or not? Right, and then I look at bigger neighborhoods and bigger neighborhoods, and I compare this to the global composition. So in other words, I want to see if local neighborhoods around sensitive cells are enriched for resistant cells or not, or are they just sort of randomly distributed? So you do this for different values of R. This is work, by the way, of Kelsey, who I mentioned earlier. She's a PhD student in biophysics. And so you get a plot that looks like this. You guys will recognize this is very similar to a correlation function, but it's easier to explain this way, I think. So this plot is the fraction of resistant neighbors as a function of radius, right? And the 0 point is just the global composition. So we set 0 to be the global composition, which means negative values here mean that the local neighborhood is less resistant than the global population. So we do this experiment with no drug around. If you happen to zoom in on a sensitive cell, you're more likely to be around another sensitive cell than you would be by just picking a cell at random in the population. Right? Any idea why that might be? We have a guess, right? Cell division, right? You can apply this to humans too, right? If you look around my house, you're more likely to find my offspring than different offspring, right? Depends on how far away you go, though, from my house. Decays very quickly. So now you could do the exact same experiment, but you could add a drug. And there you see something that we think is pretty exciting. The curve completely flips. So now what this is saying is that if I'm a sensitive cells, my neighbors have to be resistant, right? And the length scale of this is something like 1 cell diameter. Okay, what we think is happening right now is that the drug is diffusing. These resistant cells are acting as local sinks to detoxify parts of the environment. This is still preliminary. But you can imagine that because of some type of cooperation between the cells, in order for my sensitive guy to survive, he has to be surrounded, or she has to be surrounded by lots of resistant cells. Okay? So we think that's pretty cool. We can build stochastic models that capture these statistics. They don't tell us anything particularly interesting yet. Now the experiment we're really excited to do now, and we don't have results on yet, but we hopefully will soon, is imagine that we can form 2 biofilms like this, and we grow them under different conditions, but we choose the drug concentration in the blue case such that our final population composition is similar in both cases. We have the same number of resistant and sensitive cells, right? And then we want to increase the amount of drug, and what we expect to happen is that for this guy, for this curve, where the sensitive cells are randomly distributed and more close to each other because of cell division, we expect them to be depleted, right? Because they're not neighboring these resistant cells that you expect. And we think that in this case, maybe the drug will have little effect. Right? So maybe I can report back on this experiment soon. Our point is that we think spatial positioning matters, not just in the formation of the biofilm, but in its response to future drug treatment. Okay? So there are a lot of unanswered questions about this work, right? How do we choose, for example, the drug dosing over time in a smart way, right? Just from an engineering perspective or a math perspective, how would we choose it? We want to choose it, if you use too much drug at the beginning, the biofilms don't grow as much, but they form these really sort of spatially structured communities that may be much more resilient to drug in the future. So there's an optimization problem here we're trying to get our heads around. You could also say, does this type of organization change with Z position, right? So if the drug really does have trouble penetrating, you can imagine that maybe near the top we see this spatial structure, but as you go farther down, you don't see that spatial structure. What I showed you before is just a spatial average over Z. Okay? You also could ask things like, what's the role of cell shape, right? You could imagine doing this with E. coli or another bacteria. That impacts the packing density, so it's going to affect how these cells are together and sort of the length scale of these effects. What happens if we dial the enzyme production up or down? Something we're trying to do, put it on a inducible promoter, so we can dial it up and down and see if we can change either the length scale or the large scale spatial structure of this. Okay, the model predicts that predicts that there's a sort of a critical coupling where you get these long length scale behaviors and structure. Okay? We don't know the answer yet. So just a quick summary now, and then I might spend a few minutes on the final thing, but a couple things I'd like you to remember. Population density modulates drug efficacy. Doesn't seem to be particularly rare. We can capture this effect in a single parameter, right? It's kind of nice. We don't need to know the mechanism to get to measure this parameter. Once you measure the parameter, you can build mathematical models that make predictions, right? Even without knowing the mechanisms. One of those predictions is that treatment outcomes can be bistable. Okay? I also showed you that in this sort of high density limit where biofilms are forming, cell wall inhibitors are promoting biofilm formation at low doses. There's some trade-off between cell lysis and drug efficacy that's causing this. You can think of this in terms of this phase diagram. It's basically how much as a function of drug increase, how much lysis versus cell killing, sort of which one wins out. And then in the really high drug limit, what I've shown you is that sensitive cells, first of all, they survive way above the drug limit where they normally would as long as resistant cells are around, and then they appear to have these local neighborhoods that are enriched for resistant cells. Okay? So I'm going to end with one more thing. This is ongoing. It's also very preliminary. Several people in my lab are working on a problem that's on a longer term scale. And so these are really evolution experiments. And the question is, what if the environment, and for us this environment is the drug, what if the drugs are changing over time? So you're, for example, cycling the drug over time. And the question is, and this is a question that's gotten a ton of attention in both cancer therapy and antimicrobial therapy, is how would you choose the optimal drug cycles to minimize resistance? And it turns out this is a really hard problem for reasons that most evolutionary biologists would have predicted. When you evolve resistance to drug 1, and that increases over time, you may also evolve an unexpected or a collateral resistant to a second drug, right? And this happens somewhat frequently. Another thing that happens that gives you some hope, though, is sometimes as drug 1 resistance increases, the resistance to drug 2 actually decreases. Okay? So this, again, is not something surprising. If you evolve to one environment and you switch to a different environment, you can actually be hurt, right, in that new environment. You've optimized yourself for an environment you're no longer seeing. So in the case of antibiotics specifically, this was first reported in, I think it was 1952, and I can't find a single mention of it until 2013 after that, right? And in 2013, there was a group that started to ask questions about what does this look like on a large scale? Are these things common, right? And so basically where I think we are now is there are a lot of people interested in this question, and one of the reasons for that is that a lot of the questions are unanswered. Are these collateral effects common if we evolve to particular antibiotics? Are they repeatable? Can we use them to design drug cycles? Is the evolution and the genetics so complex that we essentially have no prayer? Right? This is the question we're trying to grapple with. So we did a lot of experiments. This is work from Jeff. He's a PhD student in biophysics in my group. And we did lab evolution experiments. It was just serial passage over time. We start with a small population of bacteria on day 1. Each day we dilute that we add a drug. Each day we dilute that population by some fraction, and we grow them up the next day, and over time we gradually increase the drug. So the selection pressure is increasing over time, and the resistance is also increasing over time. And then at some time point, which we determined somewhat arbitrarily at this point, we take and isolate our mutant, and we measure a dose-response curve. So basically the optical density relative to untreated cells after some time period, essentially the growth of the population as a function of the drug. And what we care about is at least for now, basically just one parameter, the IC50, which is the half maximal inhibitory concentration. The concentration of drug where growth is half what it would be otherwise. And then we repeat this. We test it against 17 different drugs, right? So we have this library of drugs. So we do this evolution, we test against all 17 drugs, and now we have the response of a mutant grown in response or selected on drug A to 17 additional drugs. Okay? And then Jeff repeated this whole thing 17 times, once for each drug. Right? So really an amazing experimental achievement, and a great, he was able to do it actually quite quickly, but it was somewhat of a leap of faith because we're still working with how to deal with the data. But I think it's cool. I told him it was courageous to do this. And we think we found some cool things. So when you do this experiment, you can condense this data set into something that looks like this. On the x-axis is the selecting drug. On the x-axis is the drug we use for selection. I've just numbered them 1 through 17 arbitrarily. On the y-axis is the drug you're testing against. The color map then indicates how much the IC50, how much the drug resistance of the mutant has increased or decreased relative to the wild type. It's on a logarithmic scale, so it just means a factor of 2 increase or decrease look the same, right, above or below 0. So if you have 0, that means the resistance is the same as the wild type. If it's blue, you have resistance. If it's red, you have sensitivity. So a couple things to notice. One is it's not surprising that when you apply a selecting drug, you always get resistance to that drug. Right? That's the diagonal. Nothing interesting there. You do see some different levels of resistance. Another thing, though, that I want to point out is that the large collateral effects are not particularly rare. Right? This goes to a factor of 2 on a log scale. So this is a factor of 4 change in the IC50 of the drug. Right? So these are relatively big effects, and there's a lot of them. Right? I pointed out some red ones and some blue ones. So the next question we asked is this repeatable or not? Right? We did this once, right? But you know how evolution goes, right? So you got to try this many times. So we started just by repeating it 4 times. And then we looked to see how repeatable are these collateral sensitivity profiles across different mutants. And this is just one of the profiles. These are all the drugs tested. It's tested against each of these 4 of the different mutants. So what you can see is, in this case, they were selected by daptomycin. All the replicates evolved resistance to that, right? But the replicates can look very different in some cases, right? So this is not particularly surprising, but we wanted to sort of get an idea of how big this stochasticity was. And then once we had this, we were like, ooh, this looks, it looks very complicated. Is there some sort of hidden structure in this data, right? And so we thought of each of these profiles as an n-dimensional vector, right? So I have a vector in some very high dimensional space. And each mutant falls somewhere in this space. And then we plot all our mutants and we use, some type of sort of hands-off algorithm to say, are there sort of clusters in this space where things seem to fall? When we look by eye, it's hard for us to tell, right? We look at this matrix, you can stare at it for a long time, it's hard to pull out structure, right? But you can do something like this very easily. So we use something that's called hierarchical clustering. Some of you may be familiar with it. As a metric, just for completeness, we use the correlation between profiles, and the linkage criteria is the mean average linking, or the mean average linkage cluster. And essentially the result, which I'm just going to say quickly, is that the mutants do tend to cluster into classes. And if you sort of zoom out and you look at the major classes that they cluster into, you find that they correspond exactly to known drug classes, right? Or almost exactly. So this gives us some hope, right? The RNA synthesis inhibitors, all the mutants for that go together. We look at them, they look quite different, right? This type of thing holds even if we take out the selection, the drug you're selecting to, because you could say it's just pulling out the drug you're selecting to, right? So this gives us some hope, right? It says there's some structure to this data. And so the thing we're looking at now, and I'll tell you the way we're thinking about it, and you can tell me what you think, is can we use this data, or data like it, maybe much more data, but data like this to predict cycling protocols. And the way we're doing this is what's called a Markov decision process. And the way this works is I consider at each time step that the system is in some state. And I define the state of the system by the collateral sensitivity profile. So that's the state of my system in this very high dimensional space. But also it has the current drug that's being applied. So if you give me the collateral sensitivity profile and the drug that's applied, you've given me everything I need to know about the current state of the system. This is a Markov model, which means the next state only depends on this current state, but it depends on it in a probabilistic way. And so we allow the current drug to determine probabilistically the sensitivity profile at the next step, right? And you can, so essentially you're rolling a dice, you know the current profile, you know the drug, you roll some dice, and in a stochastic way you determine the next profile. And the key of a model like this is that you infer this transition matrix from the evolution data. Right? Now we can argue about how exactly we should infer it, we don't know yet, we've done it some simple ways, right? But you can imagine that roughly I know from our data, or at least I can argue, that I know with some probability how often applying drug K gives me resistance to drug J, right? At least in some probabilistic way, right? So once you do that, then at each step you have to make a decision, and the decision is which drug is next, right? And the way I make this decision, the criteria for optimizing this decision is to say, I want to maximize the total amount of collateral sensitivity over time, right? So I wanted each step to be very, very sensitive, and I want the sum of all those sensitivities to be as high as possible, with the thought that maybe this could slow down evolution resistance because you're constantly flipping to a more sensitive drug. And luckily, this is a solved problem, right? Markov decision processes, there's tons of literature on this. So once we convert our data to a form that we can use it, it's a very easy problem to solve. And the solution looks like this. It's called an optimal policy. And the policy basically says, if my current drug is 1 through 17, that's on the x-axis, the optimal next drug is whatever on the y-axis, okay? So this is the drug I should pick next. And you notice I gave two solutions here. The blue solution optimizes the next step only. So it says at this step, I want the next step to be as collaterally sensitive as possible, right? But that's sort of a short-term decision that doesn't always win out. So luckily, in this theory, you can do what are called infinite time horizons. And you can also optimize long-term behavior. And one interesting thing is that the local solution, the solution you would pick if you only care about the next sort of epoch, can be very different than the longer-term solution. And so I can just very quickly show you what the policy is doing. So this is a plot of just the average level of collateral sensitivity for each selecting drug. So given the way we chose our transition matrix, for each selecting drug on the x-axis, I give you, say in a column here, the average collateral sensitivity that's likely on the next step, okay? And so for example, this column tells me if I'm selecting with drug 16, this is the average collateral sensitivity to every other drug that I can expect from that selection process, okay? And when you do this, I've marked it just for comparison, I've marked the minimum value, the most sensitive value in each column. I've done that so you can see sort of what this algorithm is doing. So if I'm selecting with drug 16, the highest sensitivity on my next step would be expected to be to drug 12. That's what I'm saying, right? And so now if you go back to these optimal policies, the instant gratification policy, the blue, is essentially just finding the minimum here. It's flipped upside down because my numbers go 1 to 16 in different orders here, right? So all it's doing is saying for each drug, I want to maximize on average how much collateral sensitivity I have at the next step. Okay? On the other hand, the long-term optimal is making suboptimal short-term steps to give you a longer-term solution that's better. In other words, you're getting rid of local minima, right, or local maxima, keeping you from getting trapped there. So you can translate these policies into optimal drug cycles, starting with any drug. So this is starting with drug 1. This is optimally how you would cycle over time based on our data. If you start with a different drug, the cycle can end up looking very different, right, based on these sensitivity profiles. Oh. Okay. It's okay, it's just almost there. It's like almost the last slide. Okay, and so your cycles will also depend on whether you care about this sort of instant gratification, short term, or the longer term solution, right? Now you can also simulate this process given the transition matrix that you have, and you can see that the instant gratification on this axis, by the way, is just collateral sensitivity with large collateral sensitivity at the bottom. The instant gratification cycle will quickly do very well on the first step, but then over time does much worse, right, than the long-term optimal. So a quick summary here, collateral effects are common, right? That's what we've seen so far. The evolution is complicated, right? Nobody's surprised by this. But there do seem to be some statistical patterns in this data, and we're hoping that you can use this to find optimal drug cycles. We think we at least have a way to think about it. So there's, of course, a lot of unanswered questions here. There's a lot of ongoing work. Please, let's talk anytime in my email. I have a modest Twitter following of like 100 people. So follow me if you want. And then thanks to all the members of my lab. I didn't get to talk about Zia's work. I talked only briefly about Max, some Kelsey, Jason, Jeff, and Wynn's work, and funding sources. So thanks again.
Mm-hmm. And that makes sense, and you showed it, sort of explains the data as well. But it seems very counterintuitive to me that cells would benefit from sort of going towards whatever, running towards the dead cells and attaching themselves there. So is there something missing? Is there something more? You mentioned briefly in passing that the concentrations of the antibiotics are actually quite low.
Right.
Does that play a role in there?
So it's a good question. I don't know the exact answer to it. Our thought has sort of been that maybe, you're right, I don't see a reason why sort of adding extracellular DNA would necessarily increase the on rate of cells, which is, but you can imagine it might slow down the off rate. Well, maybe mathematically we could consider looking at it like that.
From an evolutionary point of view, it seems like a really bad idea to go hang out there all the dead things.
I agree. Yeah, I don't know the answer to it. I think it's a great question. I don't know the answer.
Think about where these this is a drug that lives in the intestine, and it doesn't want to be flushed out. If it's flushed out, it ends up in an environment that's
Awesome.
a suitable one for the point of view of the endopockets. So if you form a biocell that you attach to the intestine, this is an adaptation that will...
Bioinformation itself, I see, is sure, it's an adaptation, but as this particular mechanism
Yeah, you're saying why evolutionarily would you couple death
Yeah, yeah.
to?
I mean, if, for example, the biofilm is protective,
Mhm.
then it does make a lot more sense.
Yeah, yeah, yeah.
Because the concentration would be lower in that.
Mm-hmm.
In part because there are protections.
Right. So I know there's some thought now, and I think there was a paper that I just saw in the archive recently, that this extracellular DNA actually can play a coding role. That cells are taking it up and using, sort of, again, evolutionarily, you might argue, yeah, but this is the DNA that didn't work so well. But
That's also going to depend on the
true.
But one thing you didn't mention was quorum sensing molecules.
Mm-hmm.
And what is the interaction with that
Mm-hmm.
But you might be just looking at some kind of particular molecular interaction.
I agree. I would guess in some of the cases with the density-dependent growth in liquid cultures, we're almost certainly seeing in some cases some type of quorum sensing. What's quorum sensing response now? Again, what's sort of interesting is that if you take away the drug, you don't see much physiology difference. That, of course, doesn't mean that there's not already cell signaling going on. Physiology difference based on our measurement.
Can we get Cheryl to stick around?
Yeah. Thanks.